One of the peaks of the summit events that we had in 2018 and also we have this year is the $1 million prize for the best research we could identify in reproductive sciences and women's health that were submitted to at our competition. The winners of the 2018 prize are Yaakov Barak, Miriam Hamburger, and Henry Sukov. Their title of the research was Placental Origin of Congenital Heart Disease. Dr. Barak is an associate professor of OB/GYN and reproductive sciences here at Magee-Womens Research Institute at the University of Pittsburgh. Miriam Hamburger is a professor of biochemistry and molecular biology and the director of Precision Medicine Disease Mechanism Program at the University of Calgary in Canada. And I'm very happy to see Miriam's face here on the monitors. Welcome, Miriam, from Calgary. And Henry Sukov, who's a professor of bioregenerative medicine and cell biology at the Medical University of South Carolina and Smart State Endowed Chair in Biofabrication. I'd like to invite Yaki and Henry and also Miriam virtually to join us for the presentation.
Congenital heart defects represent a significant burden in the human population. Congenital heart defects are the largest category of congenital birth defects in the population, affecting just under 1% of all live births. In addition, congenital heart defects underlie a large percentage of fetal demise. The combination of high frequency and the requirement for sustained clinical intervention shortly after birth and then for the rest of life represents an enormous economic burden to the healthcare system. These are some of the congenital heart defects that we have studied in this project. Ventricular septal defect, commonly described as hole in the heart, is itself the single largest subcategory of congenital birth defect, either as itself as an isolated condition or in conjunction with other congenital heart defects such as those shown in this slide. And you'll see examples of ventricular septal defect in the slides that come up. Interestingly, only about 30% of all congenital heart defects, and including the ones shown here, have an explainable cause. Those could be either genetic or exposure to an environmental agent or some basis in maternal pathophysiology. The other 70% are idiopathic, which means there's no explainable cause. In order to close this gap in knowledge, in order to reach an understanding and ultimately to achieve prevention before these occur, it is necessary to think in new ways about the causes of congenital heart defects. And one of the issues has been that most investigators in heart development focus on heart development and have overlooked events that occur outside of the heart, but that may have a significant impact on heart development and the cause of congenital heart defects.
So the concept behind our project was born over 2 decades ago when I happened to be a postdoc who analyzed mice deficient for the transcription factor PPR gamma. And I was originally interested in studying fat tissue, but nature had a mind of its own. And these mice had placental and heart defects. This was not surprising because nature always has a plan of its own when you study genes and you find things that you didn't think about. What was surprising was that PPR gamma, the gene that we were studying, was not expressed in the heart, only in the placenta, which raised the possibility, that was intriguing at the time, that the heart defects might be caused by the placental defects. A long story short, indeed they were. When we corrected the placentas using an archaic and old school technique called tetraploid chimeras, while leaving the embryo completely mutant, the heart defect disappeared despite the fact that the gene was not present in the heart. So we named this phenomenon at the time the placenta-heart axis. Over the following 2 decades, there was a trickle of about half a dozen genes in which this has been shown to be the case as well, and the names are listed here on the right of the slide. But in general, the scientific community remained skeptical, and the concept really had a hard time getting any traction. Now, roll forward to 2018, where Dr. Hamburger, who you can see here, and her colleagues, then at Cambridge, England, published a landmark paper where they analyzed over 100 randomly selected embryonic lethal mouse strains that were available in the International Knockout Mouse Consortium. What they found was that 60% of embryonic lethal knockout mice strain had a placental defect, which was an exceptionally and unexpectedly high proportion. But for the purpose of this project, what was tantalizing was that 40% of mice with placental defects had heart defects, and most of those defects were in the ventricle or the septum, the same ones that have been shown to be part of the placenta-heart axis, the limited data that we had. Now, over the last few years, there was sort of a resurgence of the concept also in the clinic. Several epidemiological studies have been published which flesh out similar associations, primarily between different types of placental defects or placenta size and CHD, congenital heart defects in general, or specific defects like ventricular septal defects or hypoplastic left heart. And in work in progress that we are doing here at Magee, together with Doctors Katov, Parks, and Weinberg at the Children's Hospital of Pittsburgh, we are finding that newborns with ventricular septal defects have an abnormally high incidence of placental pathologies that are indicative of severe disruption to blood flow on the maternal or fetal side of the placenta. So this was a ripe time to maybe start studying the placenta-heart axis in earnest. The first order of the day is to develop more robust animal models as well as cell culture models. We need models that are more closely mimicking the human situation, timing-wise and severity-wise and defect-wise. We need models that enable us to focus on the net effect of the placenta, removing any shred of influence from the embryo itself, and enabling us to focus on the net influences of the placenta on the heart. And we also would benefit greatly from having multiple models, because then we can take divergent models that converge on one point, which is the heart, and deduce by the commonalities of those models what is the mechanism that is responsible for the influence of the placenta on the heart. So the models, the mouse models, are the core of this project at this point, and these will enable us to do mechanism. We already had a very good model, a very powerful model in hand, which is SOX2-CRE. SOX2-CRE is a strain of mice that enables us to delete genes in the entire embryo, specifically without really impacting the placenta. And if you want to check if an effect comes from the placenta or not, you use SOX2-CRE. If the defect has been eliminated, it means that it came from the placenta. If the defect persisted, it means that the defect has nothing to do with the placenta. But we needed also a reciprocal arrangement. And we need to delete genes that are completely and specifically in the placenta, so that we don't look at compound influences of the placenta and other tissues and then not know what comes from where. And the next slides will illustrate how this is being taken care of. So the first example I'm going to tell you about is RXR alpha. RXR alpha is a transcription factor whose knockout causes severe ventricular septal defects and has been studied by several groups, most prominently here by Dr. Sukov. what we suspected, because there was a sort of forgotten publication that showed also placental defects in RX alpha knockouts, we suspected that the placenta heart axis is at play here as well. And indeed it was. When we used SOX2-CRE, we were able to eliminate the defects completely. But there were some residual heart defects, so we wanted the opportunity to knock out the gene only in the placenta and see the net influence of the placenta on the heart. Enter reverse RX alpha knockout. So what we did here is inserted into the RX alpha gene a removable trap where we can use the same SOX2-CRE that we normally knock out gene with to knock the gene back to life. It was a logical plan, but there was a hitch. So this is the plan, and this is the hitch. The hitch is that there was partial read-through at the gene, which restored some of RXR alpha. And as you can see at the bottom in the Western blots, there were definitely some RXR alpha expressed, and the result was that embryos that we expected to die in mid-gestation survived for most of pregnancy. This could have been a disaster, but it has been averted because the embryos, despite having some RXR alpha, were hypomorphs. They died perinatally. And most importantly here, you can see that the trap was removed completely using SOX2-CRE, and despite the complete correction of the gene now in the embryo, the mice were still dead before birth. So we are about a few weeks away from showing you a heart defect, but we fully expect that to be the case based on the phenotypes of the full RX alpha knockout. But it is the first example, I think, that we will have a placenta-driven perinatal lethal heart defect, which is very relevant to the human situation. And now Miriam will tell you some stories that came from her part of the collaboration about mouse models of placenta-driven heart defects. Take it away, Miriam.
Okay, so to overcome the problem of trophoblast-specific knockout generation, we managed to establish in a truly collaborative effort between the team members a new transgenic line termed SOX2-FLIP that achieves the restoration of a targeted allele to a functional allele, specifically in the embryo. And this is indicated from the conversion of the blue color in embryo and placenta, which demarcates the targeted mutant allele, to no blue staining in the embryo. So the gene is restored to functionality in the embryo, but not in the trophoblast cells of the placenta, as can be seen by the retaining blue color in the placenta on the right-hand bottom side. Thereby, this line can be used to generate placenta-specific knockouts. This is a novel and most efficient way for teasing out gene function in placenta and embryo, or versus embryo rather, which is superior to all prior experimental strategies and applicable to a large repository of publicly available knockouts. On the next slide, which shows 3 mouse lines from the collection of our phenotyping screen that Yaki has shown you, that exhibited both placental and heart defects, but whose etiology remained unknown. I will report on 2 of these lines today. The third one is underway. This strategy was meant to increase the pool of available model lines to enable us to extract mechanistic links of the placenta-heart axis. Here I show you one of these lines, SSR2, for which we generated the entire cohort of constitutive and conditional knockouts as shown. And as you can see from the embryo images and the micro-CT scans of the heart, the embryo-specific knockout highlighted in that box retains the heart defects, but the placenta-specific knockout does not exhibit them. This means that in case of SSR2, the embryonic dysmorphogenesis and lethality of mutants is not due to a placental defect, but is due to gene function intrinsic to the embryo proper. This is contrary to the next line I'm showing you, which is called ATP11A. Full ATP11A mutants exhibit massive edema, as you can see in that second embryo shown. The skin is basically lifting off that poor embryo, which is indicative of heart failure. VSD and sometimes extreme myocardial wall thinning is also observed, highlighted by the red arrowheads. This entire spectrum of phenotypes is recapitulated when the gene is ablated solely in the trophoblast compartment of the placenta, shown on the far right. This remarkable finding demonstrates that the ATP11A null phenotype is primarily caused by a dysfunctional placenta, providing a new model of placentally induced heart failure. Moreover, we identified this to be the case in 1 out of 2 mouse lines analyzed to date, which were selected purely on the basis of common placental and heart defects. This provides us with an expectation that a substantial fraction, perhaps up to 50% of mutants with heart defects, may have a placental cause or at least a placental contribution. Moreover, when examining placental transcriptomes of ATP11A wild-type and mutant conceptuses, we identified a large cohort of genes that is differentially expressed, indicated by the green and purple shading. It turns out that this gene set can serve as a signature to predict heart defects, which we observe in some heterozygous embryos, and this is based on the placental gene expression pattern alone. We are currently establishing whether this signature set can serve as a predictive test for these and other mouse mutant lines. So bear in mind, this is a placental transcriptome that can predict heart phenotypes. In addition to using the mouse as a powerful model, we aimed at setting up a cellular model to accelerate the investigation of how placental trophoblast cells may affect heart development. Here we focused on the early phases of development when trophoblast cells, shown in blue, are in close physical proximity to the emerging heart, which is in that orangey embryonic area, and in the red-colored embryo on the right-hand side. And they're in physical proximity to allow for paracrine interactions. To recapitulate the scenario in vitro, we took advantage of mouse embryonic stem cells that can be efficiently differentiated into cardiomyocytes in culture. We performed these experiments either with standard control medium or in the presence of medium that was preconditioned on trophoblast cells. Indeed, we found that trophoblast conditioned medium delays the cardiomyocyte differentiation process, as can be seen by the less intense green fluorescence of a cardiomyocyte reporter gene, and by the evaluation of differentiation stage-specific marker genes such as brachyury and MESP1. Overall, we found that trophoblast conditioned medium delays the differentiation process by about 1 to 2 days at the stage of cardiac mesoderm specification. This slowdown in the progression of differentiation may be required in vivo to ensure an adequate cardiac progenitor pool expansion. We then generated trophoblast stem cells mutant for the same 3 genes that we are investigating in the mouse model in vivo to test whether conditioned medium of these mutant cells has a diverging effect on cardiomyocyte differentiation. As you can see from this graph, SSR2 null and ATP11A null conditioned medium showed no difference to conditioned medium of wild-type trophoblast. By contrast, conditioned medium from SMG9 null trophoblast releases the differentiation delay and may hence cause premature cardiomyocyte differentiation in vivo. We are currently investigating this in the SMG9 targeted mice directly. We believe that our experiments reveal at least 2 distinct stages in heart development when trophoblast has a pivotal impact: an early effect, as revealed by the cell system and SMG9 deficiency, and a later effect in the second half of gestation, as shown by the ATP11A and the RXR alpha mutant mouse models. And this later effect would not be captured by the cell system.
Take it away.
With the support of the Magee Prize, this project has accomplished quite a bit. First, we have developed tools and technologies for being able to unambiguously address the role of the placenta in fetal development. These tools are useful in our own projects, of course, and also will serve as useful tools for the entire community who's interested in fetal development and the potential role of the placenta in fetal development. Conceptually, with the ATP11A and the RXR alpha allele, we have now formally proven the hypothesis of this project, which is that placental defects alone can suffice to cause congenital heart defects. And we've given you a sense, really just a hint of where this project is moving, which is to use these genetic tools and these mouse models to reach mechanistic insights and molecular insights on how the placenta influences heart development. Our team collectively co-published a review paper that lays out some of the concepts of this project, and we are working to finalize research publications that describe the work that was presented today, plus additional work that we didn't have time to discuss. The project has already generated additional resources, and most importantly, we together submitted a multi-investigator R01 grant to the NIH to continue and to expand this project. This proposal, it turns out, will be reviewed tomorrow, so please keep your fingers crossed. And finally, we leave you with some thoughts on the applications of this project. And currently, it is impossible to recognize a case of congenital heart defects before it is too late to do anything to prevent its occurrence. The heart develops earlier than all other organs of the body, but the placenta develops even earlier than that. In principle, early imaging of the placenta or the use of protein biomarkers or perhaps transcriptomics, as Miriam presented, may allow early enough detection in time to take preventative measures. I should say early enough diagnosis in advance in time to take preventative measures. Because of circumstances that affect the formation and function of the maternal side of the placenta, recognition of women who might be at risk for carrying a baby with a congenital heart defect might lead to appropriate preventative measures even before conception. And furthermore, this work leads to new opportunities for scientific discovery about how various maternal conditions, environmental agents, and genetics, fetal genetics, influence placental function at a mechanistic level and thereby influence heart development and the occurrence of congenital heart defects.
And it takes a village to do the work that we described to you here, and on the left of this slide are the people from the 3 labs who were doing the actual work in the trenches. But it also costs a pretty penny, and we cannot be thankful enough to the Richard King Mellon Foundation for rolling out the Magee Prize that really made it a reality. And if I'm allowed to do one more grateful note, it would be to the anonymous reviewers and the voting audience 3 years ago who took a chance on a project that to that time was dismissed as at best an outlier, at worst a crackpot idea. And we hope that we repaid your trust, and we are thankful for that. Thank you.